Back, everybody. Glad you're here. Before I get started today with the discussion, what questions do you have for me regarding anything? I can answer anything hopefully answer any questions you have. What questions do you have for me before we get started? Yes, Nate. No, tests are due Friday. I changed the due dates to exams, so all exams for the entire semester are to be due by the end of the day Friday. It's updated on your schedule, so just look at your course schedule. The only day that's an exception, that it's not due on a Friday, but it's due on a Thursday, is the last Thursday of the semester. So just so you're aware of that. And I believe, so that's the only date. So everything else is a Friday. I was going off on a different tangent, but I won't even go there because I don't want to say something incorrect and lead you astray. So on Fridays. And the reason why that was done was because there's some students depending on their schedules and their flexibility, that it was really tough for them, because we first get the lecture information on Thursday to get to a testing center by the end of the day Wednesday. So then I gave a couple more days. For general A&P, same kind of thing. I extended it out because we cover our information on Wednesdays, and then I had one student in particular who said, well, I can make it to the assessment center on Wednesdays. I'm like, well, that's too late. So I just pushed it out to give people more time. What other questions do you have? Yes? Is Learning Plan 2 just on what is Chapter 29? Yes, it'll just be out of Chapter 29. Yes. I'm sorry, I was thrown for a slight loop because I'm already on Learning Plan 4 for General A&P. So when you said Learning Plan 2, I was thinking I'm going backwards, but I'm not. We're in Learning Plan 2 here today. Yeah. So Chapter 29. Yeah, it all comes out Chapter 29. Yeah. Mm-hmm. What other questions do you have? Ready to roll? Okay. All right. So in Learning Plan 2, what we're going to be talking about here, we're going to be talking about, as it says, human development, human reproductive physiology. So what we're going to focus on today, we actually spent 2 weeks on this information. What we're going to be focusing on today is what happens from fertilization through roughly the end of the embryonic period, the start of the fetal period, and then just talking a little bit about what happens during the fetal period. So really what we're talking about is generally about the first 8 weeks of a human's life. That starts with fertilization. When we come back next time, then what we're going to focus on are the changes that are occurring within the mother while she's pregnant, while she's expecting. We'll talk about labor and delivery. We'll talk about the changes that the newborn has to undergo to get used to life outside of its mother, and talking about a few other things. But that's basically what this learning plan is about. So what we're going to do, first of all, we're going to start off looking at just a broad overview of the prenatal period. So looking at this picture here, we can see a little bit of a summary here, which helps us out. It's a nice picture, actually. What it starts with is fertilization. We start with fertilization right here, and then we enter what's called the pre-embryonic period. This is a period where we're setting the stage for the development of the embryo. And then we enter the embryonic period here. And then when we get to the end of the embryonic period at about week 8, then we enter the fetal period. So it says in your notes, the prenatal period is broken down into 3 shorter periods: the pre-embryonic period, the embryonic period, and the fetal period. So the pre-embryonic period is the first 2 weeks. We'll talk about that. The embryonic period is the 3rd through 8th week. This is where we see organogenesis occur. Organo refers to the organs. Genesis means the formation of or creation of. And it's amazing what happens, because if you look at a human embryo versus a chicken versus a dog versus a pig or sheep or whatever, we look remarkably similar, but in those 4 weeks we differentiate into something that looks very different from those animals, and all animals will differentiate from each other. So it's pretty amazing. What else is amazing, by the time we get done with week 8 and we enter the fetal period, all organ systems are present. So everything's there. Eyes, ears, nose, mouth, hands, fingers, toes, arms, legs, you name it. Everything. Everything. So it just develops from this point on. And just gets larger and larger and larger until we reach birth. Birth comes at about 40 weeks. So they say from the date of the mother's last period. So I always think that's kind of funny, because they consider a woman to be pregnant before she's pregnant. Because it's not like a woman gets pregnant the day after her last period, right? That's not how it works. It's not like, my period's over, oh, now I'm pregnant. It doesn't work that way. It's usually about 2 weeks. So actually, it's kind of off. It's like 38 in actuality. But it's funny how they calculate it back to the date of the last period, and then they go from there. So anyway, so this picture does a nice job of showing you a nice summary of how that all works and how it's all set up. Okay. So this table, while it's really hard for you to see from where you are, if you have your book in front of you, you can see a little bit better. But this table is a really excellent resource, because it's going to tell you what's going on each step along the way during pre-embryonic development. So we're going to start off talking about fertilization, then we're going to talk about the zygote, and then talking about the morula and the blastocyst and implantation and all that. And so as we move on and as we go through this, we're going to go through all the things that you see here. But when you're coming back and you're studying or you're reviewing this information or you're doing your homework assignments and you want this information, here it is for you in a very nice, clear, concise manner. So make sure that you check back in on that. Okay, so let's start off talking about fertilization. Fertilization, page 1, letter A. Fertilization is something that needs to occur within a certain amount of time. So if you think back to the end of general A&P, when we were covering general A&P, general anatomy physiology, what we talked about at the end, we talked about the cycles of the female reproductive system. Does anybody remember what those cycles were? What they were called? Anybody want to take a guess? Okay, what are some organs of the female reproductive system? Let's start there. I hope you can tell me, because I've got a bunch of women in this class, and I know you know your anatomy and what body parts you have. Come on. Ovaries. Okay, good. That's one. Okay, what's another organ of the female reproductive system? What? Yes, there they are, the two cycles. We have the ovarian cycle and we have the uterine cycle. So what's happening during the ovarian cycle? Okay. Most of you girls, most of you women in here knew this at by the time you were 13, 12, 13. Come on. What happens in the ovaries? The egg is released. Right, thank you. Okay? And when the egg is released, that's called what? Thank you. Good. Ovulation. Good. Okay, good. So, remember, we have the ovarian cycle. How long is the female reproductive cycle? How many days? Good. Okay, see, we're getting there. So quiet, but we know this stuff. All right, so we've got 28 days. So halfway along, okay, what's halfway between 1 and 28? 14. Good. All right, so in the ovarian cycle, right here at day 14 is ovulation. Okay? So leading up to that, we have the development of the follicle. This is called the follicular stage. And then here we have the luteal stage. And we'll talk about that in a little bit. Okay? So, at day 14, ovulation. Okay. So 12 to 24 days after, 12 to 24 days, 12 to 24 hours after ovulation, fertilization needs to occur. So that's your window. Very, very narrow. 12 to 24 hours after ovulation, that's when fertilization needs to occur. That's the ideal window. If you're a little bit before that, a little bit after that, your chances go down significantly. So between 12 and 24 hours after ovulation, that's when fertilization needs to occur. And fertilization occurs, we don't see it on a picture here, but fertilization occurs about a third of the way along the uterine tube. So we've got the uterus, and then we have the uterine tube. Then we've got the ovary here. So about a third of the way down, that's where the sperm and the egg will meet, and then this is about where fertilization occurs. 12 to 24 hours after ovulation. So when the sperm finally reaches the unfertilized egg is actually hard to access. I mean, it's there, but it's really hard to access because the egg is surrounded by a couple of layers. One layer is called the corona radiata, the other one is called the zona pellucida. And the sperm has to get through both of those layers in order to actually fertilize the egg. So there's about 100 to 300 million sperm that are typically released in the average ejaculation. 100 to 300 million sperm that get released into the female reproductive tract. Of those 100 to 300 million, about 100 to 300 will find their way toward the egg. So picture not only like 2 or 3 sperm here, but you got about 100 sperm in the general vicinity. Maybe not all are attached or trying to get into the egg, but you've got competition. That's what I'm saying. Not a lot, but you've got some. And so the sperm has to make its way through these two layers. It has to make its way through the corona radiata first, and then the zona pellucida second. That's not what I wanted here. All right. So let's look at this name, corona radiata. Corona. What does that mean? Besides beer. Right? Because that's, I think, corona. Corona. Radiata. It means the rays of the sun. So when you look at the corona radiata, they don't show it very much here, but if you picture like the rays of the sun, if I'm drawing it out here now, maybe you'll see it a little bit, right? So it looks like the rays of a sun, all right? So it's the rays of the sun. These are cells that came from the ovary. When the follicle was developing, it was surrounded by all of these follicular cells, kind of, that come from, that kind of pamper and help with the development of this egg. And so when ovulation occurs, some of those cells come along with it, and that's the corona radiata. So that's the outer layer. And then we have an inner layer here, which is called the zona pellucida, or they call it the ZP for short. This is another layer that once the sperm gets through the corona radiata, it needs to get through the zona pellucida. If it can make it through the zona pellucida and come into contact with the actual egg itself, then we're in business. So what we're seeing here, the definition of fertilization, when you have two gametes fused to form a new diploid cell composed of genetic material from both parents. All right, so the sperm contains genetic information, and then we have genetic information. We have chromosomes here within the egg itself. All right, so we've got the pronucleus of the egg. We have the pronucleus of the sperm. The two are going to come together, and when they fuse, that's when we form what we call the zygote. So when the genetic information fuses together, that's fertilization right there. Boom. Okay? So in order for the sperm to get through this outer corona radiata, the sperm has to change the way that it swims a little bit, because all this time the sperm has just kind of been swimming along straight. But now when it comes into contact with the sperm, instead of just wiggling its tail back and forth to move forward, it's going to start to wiggle back and forth much more vigorously and violently as it's trying to sort of wedge its way in between these cells to get deep into the area of the zona pellucida. And also what's going to happen is the contents of the head of the sperm are going to start to wear down. They're going to start to break down through enzymatic degradation, and the egg has some enzymes that are helping make this happen, so that way if the sperm does penetrate and get into the egg, that the genetic contents are there and available right away. And they call that capacitation. It's the removal of a glycoprotein coat and some proteins from the sperm cell membrane overlying the acrosome. Okay. So once the sperm comes into contact with the actual egg, it gets through the corona radiata, it gets through the zona pellucida. Now it's going to come into contact with the egg, and something's going to happen. It's called, I shouldn't say called anything. What it is like a shock. It's like a signal that hardens the outside of the egg so that we can't have another sperm fertilize the egg. Why don't you want another sperm fertilizing this egg? What would be the downside to that? Why isn't the more the merrier the rule of thumb here? Yeah, you'd have too many chromosomes. So we don't want many. You want 46 chromosomes. You want 23 coming from mom. You want 23 coming from dad, and that's it. Any more is usually not compatible with life. Okay. So here we got the three phases. The sperm penetrating the corona radiata, then it's going to penetrate the zona pellucida, and then it's going to fertilize the egg. Once that happens, now we've got what's called the zygote in the upper left-hand corner here. Now, these polar bodies, we're going to talk about this next time we're together for Learning Plan 3, but the way that the polar bodies develop when, I don't want to just give you the whole lecture now because I got other things to talk about, but the polar bodies really are, it's extra genetic information. So when the egg is developing into the egg, there's extra chromosomes that we don't need, because how many chromosomes do you want in the egg? We have 46 normally, but how many do you want? How many do you want in the egg? What's that? Yeah, 23. Who said 23? Very nice. 23. So you want 23. So you're going to have way too much genetic information, because you start out. I'll give you the quick math, and then I'll clear out of it, and then we'll move on. But you see these polar bodies. I just want you to know what these are. Okay, so you start with 46, but then the normal cell division process is you double it before you divide. Okay, so 46 times 2 is what? 92. Okay. Then it splits, and then we end up with 2 cells with how much in them? 46. Okay. So you're going to discard 46 of those. That becomes one of the polar bodies. But now you got to divide off again to 23 and 23. This 23 stays in the egg. This 23 is discarded as another polar body. So that's why you see these polar bodies. You're just getting rid of extra genetic information, genetic material that the body doesn't need. So in the female reproductive system, you actually get rid of the female actually discards 75% of the genetic material, whereas in the male reproductive system, they're used. In the female reproductive system, you select one egg and you pamper it and you bring it up and you help it develop. And in the male reproductive system, it's like the shotgun mentality. Just blast with as much as you can, you know. See? how things maybe aren't so different, right, when we get old. All right, anyway. So anyway, we got the zygote. All right, so we got the zygote. That's the fertilized egg. But then what happens is we start to divide the cell. And the division takes a while. The first time it takes like a day for it to divide once, but then the divisions come faster and faster and faster and faster. And that division is called cleavage. So, as you see in your notes, page 2, letter B, it says that once fertilization occurs, the zygote undergoes one cellular division, then a series of rapid cellular divisions termed cleavage. The rapid cell divisions produce numerous cells, but the overall size of the structure remains the same. Thus, each cell stays small. It's kind of like a tongue twister if you read it really fast. All right, so anyway, we go to 2 cells, then we go to 4, then obviously we go to 8. Then we continue to develop, but we don't have the time in between divisions for that cell to grow. So you've got a fixed amount of fluid in that cell, and you're just pinching off smaller and smaller and smaller divisions of that fluid. So the actual structure itself doesn't get any bigger after it's divided several times than what you started with. And it stays within the zona pellucida yet. It's still in there. So you've got this zona pellucida that surrounds these dividing cells. Eventually you get to this stage here where you call it the morula. That's number 2 under letter B. The 16-cell stage called the morula, and then it just keeps dividing from there. Morula translates to mulberry. Has anybody ever seen a mulberry? Raise your hand if you've seen a mulberry. 1, 2. What does a mulberry look like? 3. What does it look like? It's dark purple. Yeah, multiple spherical units. Everybody here knows what a raspberry looks like, or a blackberry, right? So it's similar to that. Okay, might as well call it a blackberry or a raspberry. So you've seen what those are made up of. You've got these little small balls, spherical structures that collectively make up the berry, right? That's like what a mulberry looks like too, all right? So they call it the morula, the mulberry. It's this glob of spherical cells. So when it gets to that 16-cell stage form, then 16-cell form, then it's called the morula. So it's just a term that I want you to be familiar with. Okay. So what's going to happen now after we get to the morula stage? We're going to see an important change that occurs within this structure. See, at first this was a solid ball of cells inside the zona pellucida. What's going to happen is the zona pellucida is going to start to degenerate. It's going to start to break apart, and this cluster of cells is going to start to essentially eat its way out of the zona pellucida. It's sort of, it's grown up. It no longer needs the zona pellucida. It's growing out of the zona pellucida. What you're also going to see is that these cells are going to go from forming a solid ball of cells to forming a ball, almost like a hollow-like structure, so that the structure hollows out, and you're left with this hollow ball of cells with a little chunk of cells kind of clinging to the inside of it. So can you appreciate this three-dimensional structure? It looks like a ball, but then you got this little hunk of cells like hanging on the inside of it. Do you see that? So now you can see we have two different categories of cells. We have cells that make up the ball, and then we have cells that make up this little chunk. So the cells that make up the ball collectively form what we call the trophoblast. And then this ball of cells here, or this chunk of cells here, is called the embryoblast. Some other books call it the inner cell mass, but I like embryoblast better because it tells you what this chunk of cells is going to become. What do you think it's going to become? The embryo. Yeah, it's going to become the new human being. So the trophoblast, this ball of cells, is going to be the precursor to all the supportive structures that support the growth and development of the embryo, while the embryoblast is going to develop into the embryo itself. Between the embryoblast and the trophoblast, or I should say surrounding the embryoblast, inside of the trophoblast, is a blastocyst cavity, and that cavity will eventually be filled with a fluid. So we'll get to that in a little bit, all right? So anyway, so we've got this blastocyst, okay? So make sure you know this term, blastocyst, because the blastocyst is that hollow that hollow ball of cells made up of the trophoblast and the embryoblast. Okay? So I know we've got a lot of blast terms here, so just make sure you keep them separated, keep them straight in your mind. All right, so just to review, we start with the zygote, then we divide through a process called what? What's the cell division called? Cleavage. You got it. So cleavage occurs until we get to a 16-cell stage. And what do we call that 16-cell ball or cluster? The morula. Yes, the morula. Then the morula will begin to do what? What is it going to do? Yeah, the zona pellucida is going to start to do what? It's going to start to degenerate. And what's going to happen to that solid ball of cells? What's going to happen to it? Yeah, it's going to start to hollow out. It's going to hollow out, and then you're left with two areas. What's one area? Trophoblast. And what's that? What's that? What's a trophoblast? The outer layer, making up the hollow ball, and then we have the other part, which is the embryoblast, which eventually becomes embryo. You got it. All right, is everybody with me so far? Are we all following along? Does it make sense? What's amazing is that we're at about, I don't know, probably about 7 days right now. 7 days, and all this has already happened. Mom doesn't even know she's pregnant. Yet we got a lot of stuff going on here already. The question was, at one point, or where would you find the embryoblast in the morula? At what point does it become the embryoblast, or what point does it differentiate? The answer is that point it's not specifically noted in the book or in any text. It's somewhere between the morula stage and the blastocyst, obviously, but somewhere between the morula and the blastocyst stage, what happens is those cells will start to move toward the periphery. And when they do that, there's just one group of cells that's differentiated that will remain there and become that embryoblast. So it happens very quickly. You go from 16 to 32. So I'm guessing like one or two more divisions after the morula, that's when it's going to start to hollow out. And then that's when you're going to see that. It's always there, but those cells just differentiate as those cells at that point. So that's what kind of happens. It's great that you brought that up, because what you're going to see is that things don't get created. They simply differentiate. So you've got a group of cells that are one and the same with everything around it, and then all of a sudden they make the decision to just be different. And so they start acting differently than the cells that they used to be and the cells that are around them. Differentiation, and that's what happens again and again and again and again, this idea of differentiation. And we're going to see that the embryoblast is going to undergo more differentiation yet as we move along, and so is the trophoblast. Does that answer your question? So it's a nice timeline here. So we see that we have ovulation. (12 to 24 hours after ovulation, there's fertilization. Then we're a zygote, 8 cells, 8 cells, morula, and then very quickly after that, we enter the blastocyst stage, and then we get to this point here where the blastocyst. Okay, let's orient ourselves, because I'm just jumping straight ahead here. Let's make sure we know what our anatomy is. So here's the ovary. Okay? This is the uterine tube that runs between the ovary and the uterus itself. All right, so remember now the uterus has 3 layers to it. Inner layer is called the what? Endometrium. Yeah, you got it. Okay. The middle layer is made up of muscle. We refer to the uterus as metrium, and some people say myo for muscle. So what's the middle layer called? The what metrium? Myometrium. That's the middle layer. Yep, that's the muscular layer. That's the layer that's going to be involved in labor later on. And then we have the outer region, which is called the perimetrium. So just remember those. All right, so we have the uterine cavity here. This is the, let me clear this away because we're getting a lot of junk in here. So this is the uterine cavity right here, okay? So at about 7 to 10 days, the blastocyst is going to enter the uterus, and it's going to start looking for a place to implant itself. It can't sustain itself for very long without the help from mom. It needs oxygen and nutrients not only to survive, but it also needs oxygen and nutrients in order for it to grow and develop. So what we need to do is we need to get this blastocyst to implant into the endometrial lining of the uterus so that way it can start to grow and differentiate. Okay. So the top here we have the blastocyst. We see the 2 layers, the trophoblast and the embryoblast. We know that, right? So now we're going to see at about day 7, anywhere between day 7 and 10, give or take. We're going to see that the blastocyst is going to implant itself into the wall of the uterus. Now, what we've got here in the uterus, now remember, the uterus also has its cycle. We have the ovarian cycle. We have the uterine cycle. How many days do we have to the uterine cycle? Same as ovarian cycle. How many days? How many? 14 is half. 28 days. How often does a woman have her period in general? About every 28 days, about every 4 weeks, right? Okay, so 28 days. So, let me go out of here so I can just orient you to what's happening. Let's see here, discard. Let's just add a slide there. That's good. So I can write on this and I can explain to you what's going on here. All right. So we've got all right. So during the first 14 days of the uterine cycle, the uterus is basically building itself up. Okay, because at the beginning, at day 1, that's when the endometrium was shed off. Okay? So that's called the menstrual period of the uterine cycle. So between days 1 and 14, you're building that back up. So the woman's building back up the lining of the uterus. Why is the uterine lining building up in the first place? Must be a reason for it. What's the reason for the wall of the uterus to build in thickness? Why? Yeah, to be a host to the embryo. Because if fertilization occurs, that embryo becomes the blastocyst. That's going to need somewhere to go, to implant into. So this is called the proliferative phase, where the cells are proliferating, they're growing. Then after we reach 14 days, now we enter what we call the secretory phase. And the secretory phase is where the uterus is really nice and thick. It's plush. It's got all these uterine juices, and it's got nice blood vessels in it. Why is it built up like this? It's like luxury living for a blastocyst. But if implantation doesn't occur, what's going to happen after day 28? We go back to square one, enter the menstrual phase of the uterine cycle. This layer gets shed off, and then we start again. All right? So we've got a really nice place for the uterus, for the blastocyst to implant. So keep that in mind. Let's go back to that picture because I just want to play this out. It's no secret or no mystery as to why the ovarian cycle works on a 28-day cycle as well. And guess what? Ovulation occurs right at day 14, which is the time when we go from the proliferative to the secretory phase. The timing works out perfectly because then we're right smack dab in the middle of the secretory phase when that fertilized egg should be getting to the uterus. So timing should be perfect. If ovulation occurred here, we'd be screwed, right? Because there wouldn't be any place for that blastocyst to implant. It's natural design that when the uterus is at its thickest, most rich state, that's about the time when, lo and behold, a fertilized egg, the blastocyst, will be happening to be coming by the neighborhood, right? So keep that in mind. So here we go. We've got the blastocyst that implants itself. Now, what we see is that the embryoblast will orient itself toward the wall of the uterus. This is how it implants all the time. It will implant with the embryoblast pointing toward the uterus. Now what we see is that we've got this ball of cells here, but some of those cells of the trophoblast start to eat their way into the wall of the uterus. It just starts to digest and break down and sort of dig into, like grow roots into the uterus. So we've got the cells that still maintain that circular shape, and those cells are called the cytotrophoblast. But then we have the cells that are starting to grow roots into the uterus, and that's called the syncytiotrophoblast. I know it's a long word, kind of hard to say, but that's what it's called. It's called the syncytiotrophoblast. And that syncytiotrophoblast is going to keep eating and eating and eating and eating into and proliferating more and more and more and more. And that blastocyst is going to make its way into the wall of the uterus to the point where you can't see it except for maybe a little bump on the surface of the endometrium. Here you also see the blood vessels in the uterus, and that syncytiotrophoblast eventually is going to eat its way into those blood vessels and tap into that blood supply so that as this, what will become the embryo develops, it's able to utilize the oxygen and nutrients that are coming to it by way of the mother to help its growth and development. All right? You follow me so far? Try to take this nice and slow and talk nice and slow so you get this. Okay. Where did I lose anybody? We're still following along all right? Okay. All right. The next thing we're going to see here, if you'll notice, you don't have to tell me exactly what's going on, but what do you notice? Let me clear this off. That's not what I wanted. Push the wrong one again here. Going to play that way, aren't you? Just want to clear this slide off. That's all I want to do. All right. So we got 4 steps here. 1, 2, 3, 4. What do you see happening between steps 2 and 3? What do you see? The what's that? Yeah. We see development of that embryoblast. We see a yolk sac. We see something called a hypoblast, and we see something called an epiblast. What we see is that the embryoblast is going to start to differentiate. And so we're going to see 2 bubbles develop. I call them bubbles, but they're 2 spaces or cavities, if you want to call it that. We see one cavity here called the yolk sac, and then we have another one here, which they don't have labeled. So we have 2 cavities. I'll get back to this in just a little bit. 2 cavities develop. But what's even more important is that we've developed 2 layers of cells here. One layer they're showing in blue called the epiblast, and the other one called the hypoblast. This is what we call the bilaminar germinal disc. Bilaminar means 2 layers. Bilaminar, bilayered, okay? And some books will call that the bilayered embryonic disc, or they call this the germinal disc. Each book calls it a little bit different structure. Germinal means what? What does it mean to germinate? If I'm going to germinate seeds, what does that mean? What's that? Yeah, it's going to be sprouting growth. If I got some seeds, some bean seeds, for example, one of the things like the kindergartners do is they'll get some bean plants and they'll get a paper towel and get the paper towel wet and stick it in a plastic bag, and then they watch the bean seed germinate. It sprouts new growth. So when you think of these germinal layers, think about these layers as sprouting new growth or new life. So we've got this bilaminar germinal disc between these 2 cavities. One is the yolk sac. The other one is called the amnion. And guess what the amnion is filled with? Amniotic fluid. So here we got amniotic fluid at day 8. Pretty incredible, huh? But we have to figure out how the heck we get amniotic fluid, which is right now next to the embryo. How do we get it so that the amniotic fluid surrounds that entire fetus by the time it's ready to be delivered in an amniotic sac? And the amniotic sac is here already. It's right here. There's the amniotic sac. Well, but the amniotic sac is sitting right next to the embryo. And when we get to the fetal stage, The amniotic sac is completely surrounding the fetus. How do we get there? Okay, we'll get there. So that's what we've got. All right. So we've got the yolk sac, we've got the hypoblast, we've got the epiblast, we have the amniotic cavity, all right, and the amnion. Now, surrounding this structure, look at how that trophoblast is developed. Cytotrophoblast is still there, but the syncytiotrophoblast keeps getting bigger and bigger and more diverse. And that's because it's going to start to develop into something. It's going to develop into the precursor of what keeps this embryo and fetus alive. What keeps the embryo and fetus alive? What's the lifeline between the embryo and the fetus and its mom? Yeah, the umbilical cord. So we're going to start to see that the umbilical cord is going to start to develop, and it develops from a structure called the chorion. And that's where we're going to go in a little bit. All right. But we've got, I want to address this before we take our break. But we've got a bit of a problem here now. Because we're on the clock. We're on the clock right now. And what I mean by that is the clock is still ticking. Does mom realize that she's pregnant? We're 8 days in, 9 days in. Does mom realize she's pregnant? No, she doesn't. She has no clue. Does mom's body know that she's pregnant? We're thinking, who says yes? I see some nods. Who says no? You think you got this little thing burrowing itself into the wall of the uterus. You think mom's body would know about it. Right now, mom's body doesn't know about it yet. Part of her uterus knows, but that's about it. The rest of mom has no clue that this is going on. And if mom's body doesn't realize that she has a fertilized egg turning into an embryo inside of her, what's going to happen if her body doesn't realize that she's pregnant? Think about the uterine cycle. What's that? Yeah, it'll flush it out. So we have to have some way of keeping the wall of the uterus intact until we can send out an SOS from the embryo saying, just so you know, I'm here. Okay? And we have to do that through chemical signals. Called what? What are the chemical signals of the body? Hormones. Good. Okay, good. So let's go back because we forget about someone. Because it doesn't look very interesting, doesn't look very anything at all. Well, I don't really have a good picture of it, but I can see it here on this picture. Okay. So, remember I said that we have 14 days to the ovarian cycle? Right smack dab in the middle was what? Ovulation. Okay? So we have the first 14 days. Now I'm thinking the uterine cycle. Now I forgot what I mentioned for the first 14 days of the ovarian cycle. It'll come back to me. Follicular. Okay, so we have the follicular stage where we're developing the follicle so it can become the ovum or the unfertilized egg, okay? The mature Graafian follicle that gets ovulated. But the second 14 days are called the luteal, is called the luteal phase. Why is it called the luteal phase? What is that referring to? Do you remember? Or do you know? It has to do with the ovary, and it has to do with after ovulation takes place. There's a reason why we call the phase the luteal phase, because the luteal phase is supremely important. What's left after ovulation occurs on the surface of the ovary? What is the structure called that little pit, that little scar that remains after ovulation has taken place? Does anyone remember? Starts with corpus, ends with luteum. It's a corpus luteum. Okay? We forget about it. We don't think about it. But it's called the corpus luteum. What's the second stage of the ovarian cycle called? Luteal phase. So what does the luteal phase have to do with? The corpus luteum. Yes. So here's what the corpus luteum does. The corpus luteum secretes chemical messengers called what? Hormones. What are the hormones of the female reproductive system? You know one of them for sure. Estrogen. The other one is progesterone. So the corpus luteum secretes estrogen and progesterone in high amounts. And when the estrogen and progesterone is secreted in high amounts throughout the body, that's a chemical messenger to the uterus saying, keep the endometrium intact, just in case fertilization occurs. Will the corpus luteum release estrogen and progesterone regardless of whether the mother becomes pregnant or not? Will it? Yes, the answer is yes, it will. Because it's not like when a woman isn't pregnant that after 14 days she goes right into the menstrual phase, right? It's not like she has shorter periods or shorter intervals between periods when she's not pregnant. It's always 28 days, normally. So that means that for 14 days, the corpus luteum, which is right over here, the corpus luteum will secrete estrogen and progesterone, maintaining the wall of the endometrium, maintaining that lining just in case. But after that 14 days of the luteal phase is over and we get to the end of the ovarian cycle, what's going to happen? The corpus luteum degenerates. What happens to the estrogen and progesterone levels as it starts to degenerate? Are they going to go up or down? What do you think? They're going to go down. And when they go down, that's the signal for the woman to enter the menstrual phase of the uterine cycle. Okay? So, if estrogen and progesterone keep that endometrial lining there, what do you think that little blastocyst needs to start to do? It needs to start secreting what? Hormones. What hormones? I mentioned them before. The same two. Estrogen and progesterone. Yes. So the chorion is the precursor not only to the umbilical cord, but the chorion is the precursor to a structure which connects the umbilical cord to the mother's body. What's that called? The placenta. You got it. So the placenta will begin to secrete estrogen and progesterone on behalf of the embryo and the fetus, depending on which stage you're in. The placenta will take over the job of secreting estrogen and progesterone. So as long as there is a sufficient amount of estrogen and progesterone, what's going to happen to the endometrium? It's going to stay around. That's it. Okay. So remember, the corpus luteum, after ovulation, the corpus luteum secretes estrogen and progesterone so that the endometrium stays intact just in case implantation occurs. If implantation occurs and things go as planned, that blastocyst is going to start to develop structures that become the placenta, which will take over the role of secreting estrogen and progesterone, thereby keeping the endometrium intact. If things don't go as planned, if fertilization didn't occur, then there's no placenta. There's no taking over. And so as the corpus luteum degenerates, down go the estrogen and progesterone levels, and then she will enter the menstrual phase of the uterine cycle, and everything starts over again. Does that make sense? So that's kind of what's at play here. So we have to kind of think of what the scenarios are, but that's what's happening. The woman's body doesn't know when implantation starts that she's pregnant. The only way that she's going to know, quote unquote, know, is when that placenta, or the chorion, really, the chorion starts to release estrogen and progesterone. Last thing before we take a break. The chorion also secretes its own hormone called human chorionic gonadotropin, Gonado, 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 tropin, Tropin, all one word. Human, next word is chorionic, 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 referring to the chorion, human chorionic gonadotropin, HCG. Who's ever heard of HCG before? A couple hands. Why you heard of HCG? Yeah, it tells you you're pregnant. Yeah. That is what the pregnancy test will detect in your urine. So when the chorion starts to develop, then the woman will start to secrete HCG, or human chorionic gonadotropin. And after a few weeks, those levels go way up. And so when mom misses her period and she's starting to get suspicious, she takes a pregnancy test and it says positive. It's positive because the chorion has been producing human chorionic gonadotropin, or HCG. So the chorion not only leads to the placenta, which secretes the estrogen and progesterone, but the chorion also secretes HCG, which is going to tell mom when she takes the pregnancy test that she's pregnant. See. So we're going to take a break. We'll take 10. When we come back from the break, we'll look at the chorion and how it develops, and then we'll continue to look at the development of the embryo and see how we go from a two-dimensional flat structure to a three-dimensional structure that looks like a human. Okay, so let's go ahead and take 10, and then when we come back, we'll go from there.